that results in gametes with too many chromosomes. I combine these together, and I now get an individual that's a diploid of 8, where the original parent here was a diploid of 4. I've effectively doubled the number of chromosomes, and if you look, I've changed the number of chromosomes within a set. So this tetrad here, let's say, represents the 2 chromosome 1s that occur in this cell, and this represents the 2 chromosome 2s. With non-disjunction, right, they didn't pull apart properly, I ended up with them all on one side. Here, let's color code them like this. That means that 2 of these are chromosome 1s, and 2 of these are chromosome 2s. Same thing happened in the other individual, and now I've got 4 chromosome 1s and 4 chromosome 2s. Well, we're supposed to have 2 of every chromosome, 1 from each parent. This individual got 2 of each chromosome type, 2 from each parent, right? 2 chromosome 1s, 2 chromosome 1s, to get 4 chromosome 1s, 2 chromosome 2s from dad, 2 chromosome 2s from mom, 4 chromosome 2s. So this really represents not just a doubling in the number of chromosomes, but the doubling of the number of chromosomes in the set. Okay? Doing okay so far? So let's push this out a little bit. If Spartina alterniflora is a 2n of 62, and Spartina maritima is a 2n of 60. Normally, I'd make an n equals 31, and this would make an n equals 30. But this one messed up meiosis. It messed up meiosis, and instead of making an n equals 31, 4 made a 4, a 62 made a 62. And at the same time, this weird happenstance, here, instead of taking a 60 to a 30, the 60 was taken to a 60 because of this non-disjunction. Now, in this state, the 2 species can produce an offspring. And this offspring would be 2n equals 122. 2n equals 122. This is sympatric speciation. We just created a new species, and this one's called Spartina anglica. Now, it's a new species because this species cannot reproduce with either alterniflora or maritima. By definition, it's a new species because it's incapable of breeding with either of the 2 parent species. So we produced a new species instantaneously through a hybridization event, right? Sympatric speciation, a mistake in meiosis that allowed us to produce a hybrid offspring that's now a new species. Estimates are about, I've seen this, like 60 to 80% of the plant species, flowering plant species on the planet arose through this kind of process. A change in the number of chromosomes, this doubling that resulted in a new species. Now, Spartina anglica, it's worth talking about here for a second, is an interesting species because it does not do sexual reproduction very well. It has a lot of trouble with meiosis. Any idea why it would have a lot of trouble with meiosis? Well, I mean, we do meiosis with 46 chromosomes, and I always imagine it's a nightmare trying to line up 23 pairs of chromosomes along the metaphase plate and not get anything tangled and get everything moving to the right side of the cell without any issues. Well, imagine trying to do that with 122 chromosomes. It has a hard time doing meiosis, and as a consequence, it doesn't produce seed very well. Okay? In fact, most of the time, the way it spreads is through what's called clonal reproduction. The plant just sends out these runners to produce new individuals, and then these connections break over time, and this allows it to colonize areas of mudflats, right, these coastal regions, very easily. Because it has extra copies of chromosomes, all things being equal, it can make more protein if it's got the raw materials, so it has an advantage over its parents in its production ability. So Spartina anglica, since the days of the colonies back in the 1600s, Spartina anglica has taken over Great Britain. It's outcompeted Maritima. It spread to coastal Europe, France, Spain. It's taken over there. Somebody got the bright idea to bring it to Southeast Asia to control coastal erosion. It's taken over there, and then it got into Australia, and now it's a really, really tough invasive species that exists in other parts of the world. This species was formed through hybridization through a sympatric speciation event. Okay, I think I have a couple of other examples here. This is the kind of mustard family, they call it. The species on the corners of this species triangle are the original species. So this one would be 2n equals 18, n equals 9. That's Brassica oleracea, what I call the awful vegetable species. Cauliflower, kale, broccoli, kohlrabi, and my least favorite vegetable of all time, Brussels sprouts. And then on this corner, there's a 2n equals 20, n equals 10. That's turnip or Chinese cabbage. And at the top there is black mustard. That's a 2n equals 16, n equals 18. Okay, now these three species are closely related, but they don't reproduce together. They're their own species. But under the appropriate conditions, they can. And the condition is you need to mess up meiosis here and mess up meiosis there at exactly the same time. And if you do that, that would be an n of 18 plus an n of 16 would give me a 2n of 34 or an n of 17. That's Ethiopian mustard. That species arose through sympatric speciation. Brassica rapa, 2n 20, 2n 18, gives you a 2n of 38, which is Brassica napus. That's rapeseed. They use it to make oil. If you look on your potato chip packages, you'll see like rapeseed oil. And then here, 2n equals 16, 2n equals 20, that's 36 2n, that's brown mustard. So these three species arose the same way that Spartina anglica did, through this very bizarre mess up in meiosis followed by sympatric speciation. Okay? All right? Okay so far? All right. So, I've been moving a little bit faster because I haven't been doing those pre-slides that I'd like to do normally with a class where I can interact with, and we haven't been doing the handing back exams and going through those, and we haven't been doing kind of you stopping me during lecture to ask questions, so we are running a little bit ahead of time. Some semesters I have run a little bit ahead of time, and when I do, I usually stick in a little bit on human evolution before we get to photosynthesis. So I'm going to go ahead and do that this semester. It's kind of nice because it will allow us to put together some concepts that we've talked about so far. Phylogenetic trees, a little bit about genetics, a little bit about speciation, and what we're going to kind of address as is the kind of the prevailing hypothesis for where do humans come from? Or more specifically, how did this species arise on the planet? Okay? So what we'll do is take what we've talked about so far and try to come up with a decent hypothesis to explain this. Okay? All right. So we can generally start with this phylogeny. There's a general agreement among the biologists that the Homo sapiens species is most related to the apes, and more specifically, the African great apes. So here, this would be one of these apes. These would be the great apes, and then these are the African great apes. So based on the phylogeny stuff that we've talked about before, more closely related is a more recent common ancestor. So humans have the most distant common ancestor with the old world monkeys, as they're called, and then another common ancestor with the apes, and then a common ancestor with the great apes, and then a common ancestor with the African great apes. So a more recent common ancestor is more closely related. Okay? Now, humans share a lot of characteristics with the apes. And these characteristics are derived characteristics. And these derived characteristics are called synapomorphies. These derived characteristics are referred to as synapomorphies. These characteristics would be traits that allowed this phylogenetic tree to be manufactured. So things that we share in common with these other apes include a large brain, no tail, erect posture, right, not walking on all fours but more upright, flexibility, hips and hands. And I think I mentioned this before when we were talking about phylogenetic constraint. As soon as you move to an upright posture, this girdle can do other things that it wasn't supposed to be able to do because it doesn't have to be load-bearing anymore. And along with upright, this kind of flexibility in that pelvic girdle too also exists. We also have more flexibility in our ankles than the other vertebrates do. So the apes share this in common. And we also have a lot of flexibility in our thumb. This is characteristic of the apes too, right? The ability to kind of move that thumb in ways that you can't move the other fingers. It has tremendous flexibility based on the way that it's been designed. Okay, so these traits are traits associated with the apes. So all of these would have those characteristics. Then we could say, well, what additional traits do we share in common with the great apes, specifically the African great apes? And what are the synapomorphies shared by the African great apes? So these would include an elongated skull, right? Elongated skull, along with enlarged brow ridges. So along here, they tend to stick out a little bit. Those are enlarged brow ridges. Short, stout canines. Right? These canine teeth tend to be shorter and stouter, not as long and pointed as in the other apes or monkeys. The premaxilla, that's this bone here. Differences in the premaxilla associated with the skull, and then fusion of certain wrist bones, and that fusion again is associated with the flexibility. And the African great apes also have enlarged ovaries and mammary glands along with reduced hairiness. That reduced hairiness tends to be in certain parts of the body. So if you look at like the gorilla and the chimp here and the bonobo, right, there's reduced hairiness in a certain part of their face. I think some of the African great apes also have kind of a hairy butt, or a hirsute. No, hirsute is hairy, so it would be bald, right? No hair on their butts. Okay, so these are the characteristics that are being used to manufacture this particular phylogenetic tree. Okay? But this question mark here is here because all of the African great apes share these traits in common. It's tough to find any traits that are similar between humans and bonobos, or humans and chimps, or humans and gorillas that would allow us to figure out who is more related to whom and build a phylogenetic tree that would be appropriate. So here, what we've got is the use of particular protein molecules. So this particular tree is based on serum albumin. So this is based on protein similarities. Okay? And based on similarities in serum albumin, we have the same problem. We can still build a phylogenetic tree, and because of the molecular clock, we can put a timeline on this. But again, it's pretty unsatisfying because it looks like, based on the similarities in this protein, humans, chimps, bonobos, and gorillas, you can't tell who's more closely related to whom. Are we more closely related to chimps or gorillas based on this protein? You can't answer that question. So you can build